So good evening everyone. So I think I will just start my lecture here and let others join afterwards. So I will just start presenting the lecture. Uh, so I hope the slides are visible. Mm. Okay, so good evening everyone. So today I'm going to take the week 6 section on the topic cell culture technologies that is conducted by NPTEL for my TAship assistants. So I will just start with a reasoning of a previous question that was asked in the last section itself on last Sunday. So it was asked that what is the role of carbon dioxide and what is the importance of carbon dioxide in cell culturing. So basically carbon dioxide is especially if you know that it is particularly a gas that is being expelled out of the cell, right? So basically the cell carries out several metabolic reactions as well as it also helps in the it also undergoes respiration, right? So byproduct of this particular process is carbon dioxide basically. So in this case you know that the carbon dioxide, the carbonic acid buffer system is a particular or important buffer system that is involved in our body as well as in other organisms also, and these are involved in maintaining the pH regulation, right? So suppose that this is the cell culture system, and also in the cell culture there is carbon dioxide that is being produced by different metabolic process. So this carbon dioxide can combine with water to form carbonic acid, and this carbonic acid can later split into, in equilibrium condition, on with respect to the equilibrium condition, this carbonic acid can split into proton as well as a bicarbonate ion. This bicarbonate ion can later split into one more proton and a carbonate ion. So this process happens in the base of equilibrium status that is present within that particular medium there. So sometimes what happens is that the atmospheric carbon dioxide that is present outside the media can dissolve within the media, and it can then form carbonic acid in the presence of water that is present in the media itself. So this particular bicarbonate ion, you can see that it can release proton as well as bicarbonate ion. So on the basis of this particular system has been used as a buffering system there. So suppose that if the pH of the media decreases under certain circumstances, that is, it becomes acidic. If the pH decreases, then media is becoming more acidic, right? So this is basically due to presence of the, as I said before, the cell culture the cells can undergo respiration, which is a which releases the byproduct as carbon dioxide there. So within each process it happens, the carbon dioxide level increases. So the chances are that this carbon dioxide within the media can combine with water to form bicarbonic acid, and then it start to release more protons as well as bicarbonate ions. As the amount of bicarbonic acid increases, in this case, sorry, the carbonic acid increases in rate. The carbonic acid increases in this case, it can actually make the media more acidic. So in order to prevent that, the media is supplemented with a sodium bicarbonate within the media, which can split into sodium ions as well as the bicarbonate ions. This bicarbonate ion that is present within the media can later combine with these protons because of the formation of excessive number of bicarbonic acid here, carbonic acid here, which releases protons. It can combine with these protons and keep the pH balance at check at an optimum pH of about 7.2 to 7.4. So this is why the media is supplemented with bicarbonate ions there. And if the media becomes more basic, that is, the pH increases somehow because of the metabolic activities that is taking place within the media or due to any other changes, what happens is that the carbon dioxide that is provided from the atmosphere. So if you have seen that we have usually keeping the culture plate in a carbon dioxide incubator, right? So we are providing that particular carbon dioxide from outside. So this can dissolve within the media in the presence of water. It can produce bicarbonate ions. So keeping the pH check, right? The basic pH will come down into a neutral pH there. So this is how the actual buffering happens in the presence of a carbonic acid buffer there. So I just thought of repeating this explanation because someone asked the particular question in the last class there. So I thought to clarify this out. So does anyone have any doubt regarding this, or should I explain it once more?
Oh, sir.
Okay, so thank you. So I will just move on to the next question, question number 1. So for the answering the question, you can either unmute yourself or you can just type in the chat box. So coming to the question number 1, concanavalin A is a, we have discussed this last class, so I think you know the answer, protein binding carbohydrate, protein binding protein, carbohydrate binding protein, or carbohydrate binding carbohydrate, which might be
Carbohydrate binding protein.
carbohydrate binding protein, right? So basically this is the structure that of a concanavalin A that I later earlier on got from PDB. So PDB is also called protein data bank. You can get the structure of proteins, basically each and every protein that has been crystallographed or has done a cryo-EM and all. So basically it is a lectin, so which binds to which is a carbohydrate binding protein. The term lectin itself is a carbohydrate binding protein which has been extracted from jack bean or Cannavalia ensiformis. So it is basically a legume, that is peas, beans and everything that belong to this particular family. And this concanavalin A or cone, as abbreviated as cone, can bind to several glycoproteins or glycolipids, because the glycoproteins or a glycolipid contains carbohydrate moiety present there, right? Glycoprotein is basically a protein that consists of carbohydrate moiety bound to it, or glycolipids are lipids that consist of a carbohydrate moiety bound to it. So in the presence of this mannose group or glucosyl group, this concanavalin A recognizes it and binds to it. So basically, if you look into cell culture and all, cell culture aspect and all, several of the like you know that extracellular matrix like poly D lysine is there, poly D ornithine, collagen, several are there, like women etc. are there. So basically, in some cases, these extracellular materials cannot be used. So in that case, in that case of like in case of rods and cones, like rods are and cones are photoreceptors present in ice, right? So these consist of several glycoproteins that are present at the surface. So only in the presence of cone as a extracellular matrix, these rods and cones can bind to this particular extracellular matrix and then proliferate and differentiate. So even the presence of poly D lysine they do not adhere. Only in the presence of concanavalin A it adheres to it because of the presence of large number of glycoproteins at the surface. So this particularly minus binding property allows it to interact with the glycoproteins on the surface, facilitating cell adhesion and cell growth. So I hope that this is clear. Does anyone have any doubt? Okay, so yes, okay, so I will move on to the next question then. So which of the following is not a component of the defined system? First one is serum, second one is minerals, salt or vitamins, which might be the answer. Defined medium.
Option 1.
Option 1, serum, right? So serum is the answer basically. So if you look into a serum, basically it is obtained from animal blood, right? It's an animal source. So basically it depends on the body physiology of that animal or the organism that we are obtaining from, right? So it is not always constant, it will always change, and it consists of several other components like proteins, like albumin is there, hormones are there, growth factors are there. So basically based on, as I said, based on the physiology or the organism, the composition will be changing in that case. So we don't know the exact composition in this case. So whenever if you are adding the serum into a medium, like as I think like fetal bovine serum is there, which is present within DMEM or Dalbecos modified eagle medium, right? So in that case, we don't know the specified composition what is present within that medium there. So serum, if you are adding into a particular media, if you are adding a serum into a defined media, then it will become a complex media or a simple media, something like that. So complex media, not a simple media. Because or rich media, if you can call, because we don't know the composition of that media there, but we do know that it promotes the growth of organisms or any other cell culture that we have yet to be cultured there. So based on the classification, just I have given a additional note on this. So based on the classification, there are different types of media, like three types are there: a simple media or a basal media, then there is a complex media, and a synthetic or a defined media that we have just discussed. So simple media or a basal media composed of the basic nutrients only. So basically, if you need to grow a simple organism, or a multiple species of an organism, that you just provide a media which consists of a not that much of a composition there, like a little amount of like media there, or any other type of nutrient there that is sufficient for that particular organism to grow, right? So one such example is nutrient broth and nutrient agar there. And but we do know that it provides carbon and nitrogen sources, which are obviously what is needed for the survival of that organism. So the composition is not that much complex in case of simple media, so it is also called as a basal media there. Coming on to the complex media, we do know that there are variety of ingredients that are present in there. Some examples include extract from, like yeast extract is there, meat or plants is there. Basically it's actually like combination of different what you say nutrients there, or any other components that promotes growth of that cell culture there. So if it takes two different simple media or any multiple simple media and put it together, then it becomes a complex media there. So in that case also we don't know the exact composition, what it is defined of, how much of a particular substance that we are using or seeing there is present there. So that's why it's called as a complex media, because it consists of multiple nutrients or ingredients there, and we don't know the composition of that media. So basically this media can also be called as a rich media, because it consists of all the nutrients or for that particular organism to grow to. Rich media, from the name itself you can call it as, because it consists of all the substance that the component or culture needs to grow it. And then comes the synthetic or a defined media there. So in the name itself you know that it is something that is defined. You know what makes up the particular media there. So if you have a substance A, you are adding a substance A, and you know how much you are adding to that substance A or adding to that media. If you are adding a substance B, you weigh it out, and so that you can know what is substance B is, and what kind of how much weight or how much gram of that particular component that you are adding to it. So that's why it's called as a synthetic media. So if a synthetic media or a defined media is only used when you are to grow a particular species of an organism, rather than growing a multiple species of the same organism there in this case. So I hope that's clear there. So anyone has any doubt there? This was for general information only.
No, sir.
Okay, so I think I will move on to the next question then. So question number 3. Sorry that I forgot to arrange the slides there. So coming to the question number 3, trituration in cell culture is a chemical method of cell dissociation, mechanical method of cell dissociation, chemical method to promote cell attachment, or mechanical method to promote cell attachment. Which might be the answer?
Option 2.
Option 2, mechanical method of cell diffusion. Yes, you are right. So trituration in cell culture is a mechanical method of cell dissociation. So basically this technique includes repeated passaging of cell aggregates, because once the cell grows you need to grow it in large number, right? So if you need to have high number, then you have to do a passaging, or if you need to grow the cell for a long period of time, you need to do passaging from cell culture, right? So repeated passaging is one thing, and another most important thing that involved in trituration is when you are putting up a cell culture, right? So you are putting up a pipette into that. You cut a small at the tip of the pipette there, then you are inserting the pipette there, and then you are pipetting it up and down there. so that the cell will go into the pipette tip and then it will come out. In that process you are providing a mechanical stress or a mechanical shear force there. So in the presence of that mechanical stress or a mechanical shear force, the cells that are forming that tissue get separated out. So sometimes it is not that efficient, sometimes it is efficient because the chances of cell getting like ruptured over time is more in this case. In case of like a fetal brain tissue, trituration is preferred. In case of adult brain tissue which is more complex or aggregated in nature, we cannot do that. So in case of fetal tissue, brain tissue, like we are obtaining a brain tissue from a fetus of a rat or mouse, whatever you are looking at, and then you are like putting it in a, like you are mildly vortexing it and then putting it into a pipetting it in and out, then you are providing a mechanical stress there, right? So this effectively breaks them into smaller compartments, typically single cells or small clusters. So sometimes it is also used in case of enzymatic dissociation to optimize cell yield and viability. As I said, right, in case of adult fetal brain cells and all, you have to first do an enzymatic dissociation there in the presence of different enzymes that we will discuss later, and then you are doing a trituration, that is providing a mechanical stress using a pipette there, because it increases the efficiency of that particular experiment there. So I think this answer is clear. So does anyone have any doubt? Should I move on to the next question? Okay, so I think it's quite understandable. It's a basic question there. So I'll just move on to the next question. So coming on to the question number 4. So papain is dash derived from papaya plant that is used as a dash in cell culture. First option, enzyme, chemical method of cell dissociation, enzyme, mechanical method of cell dissociation, and extracellular matrix molecule, chemical method to promote cell attachment, and extracellular matrix molecule or a mechanical method to promote cell attachment, which might be the answer. Papain. Okay, so the answer to the question is papain is an enzyme derived from the papaya plant that is used as a chemical method, or most probably and most adjectively as a enzymatic method of cell division in a cell culture, because like it is most probably an enzyme, right? Basically from this you can see that it's 15 protease. It consists of certain catalytic residues there at the enzymatic surface. So it is basically a protein that provides it as the protease activity there, and is obtained from papaya, which is also called, the scientific name is Carica papaya there. Papaya's scientific name is Carica papaya there. So basically it is an enzyme derived from the papaya and is used to promote the chemical method or a enzymatic method of cell dissociation there. So basically if you look into how the cells are kept together, right? You know that the cells are kept together in the presence of an extracellular matrix. So proteoglycans are there, vimenins are there, several other collagen is there, several extracellular matrices are there. So these are all basically proteins, right? So basically if you are providing a particular protein that could or a protease that could degrade out this extracellular matrix without affecting the cell culture or an individual cell as a whole, only affecting the extracellular matrix, you can basically break them down into individual cells there. And this particular enzymatic method is useful when you are doing a cell culturing and all, and if you want to incubate a cell culture into a cell culture plate. So basically papain is one that kind of a protease, so obviously a cysteine protease there, and is used in this kind of process there. So in this case, so that the cells, you know that the extracellular matrix have to be degraded to achieve this, but in order to prevent or reduce, in order to prevent the reduction in the viability of cell, because we don't want to get any cells getting affected there, you have to use a low concentration of the particular enzyme. Whatever enzyme you are using, a papain or a collagenase, whatever it is, you need to use only a low concentration of this enzyme so that it doesn't affect the structural integrity or any other process of the individual cell. It should only affect the or degrade the extracellular matrix there. So papain is used in this process in lower concentration there. So rather than compared to traditional method like titration and all, this method is crucial for maintaining cell integrity and functionality, provided that it is given in low concentration at an optimum concentration where this degradation happens. So I think this question is clear. So anyone has any doubt?
No,
So
sir.
I think I will just move on to the next question then. Coming on to question number 9, like papain is a dash that dash. For the first option include carbohydrate cleaves tyrosine tryptophan bonds. Second option proteinase cleaves cysteine bonds. Third option carbohydrate join alanine bonds. Fourth option proteinase or joins lysine bonds. Which might be the answer?
Option 2.
Option 2 is a proteinase which cleaves cysteine bonds, right? So basically it's a cysteine protease. From the name itself you know that is cysteine protease, right? And it is also called as thiol protease because in case of cysteine you know that the sulfur group is present there, that's why it gets a name. So if SH group is present there, then if a protein that cleaves these SH groups there, then it is called as a thiol protease or a cysteine protease. So it is a hydrolase enzyme. Hydrolysis happens in this case. So in that case, when hydrolysis happens, the peptide bonds get broken and then it gets individually cleaved out there. So it uses a cysteine residue that is present on the surface there. Basically, it is a catalytic triad there. So as I said, there are certain amino acids that are present within the cysteine protease there. So it exists as a triad, basically a group of 3 amino acids there, and this 3 amino acid performs the catalytic activity of that particular protease there, the cysteine protease there. So these triads include cysteine, histidine and asparagine or aspartic residue in some case, but these most commonly these 3 amino acids are involved there. So this cysteine thiol, this group act as a nucleophile there, and a nucleophile means that it has the capability to give out electrons, right? Electrophile meaning that it attracts electrons, nucleophile means that it gives out electrons there. So or it has the tendency to attract protons to itself. So that attacks the peptide bond in that particular substrate, facilitating protein degradation. So there's a basic sorry. So I think I will just present by this. I think there's an issue with the, I don't know. So basically, as I said, basically it cleaves the extracellular matrix there, so it has the ability to do that basically. And also in the cell culture, papain, as I said, that it cleaves the extracellular matrix there, which are crucial for maintaining the tissue structure there. And it also facilitates the dissociation of cells from the surrounding matrix, allow for generation of single cell suspension necessary for experimental applications. So I think this is quite clear. So does anyone has any doubt?
No, sir.
Okay. So sorry for the in between. I think there was some network error in there. Okay. So coming on to question number 6. So if I have 1 lakh cells, what is the maximum number of 18 millimeter round cover slip that can be plated at a density of 50 cells per millimeter square? So this is basically a mathematical question. So sorry for that. So presentation I cannot show there. So answer is also there. So I will just explain the answer there basically. So the answer to this question is 7. So there are 4 different options: 5, 7, 11 and 13. So answer to this question is 7 there. So basically, if you look into the question, you know that they have given that how many number of cells that are present within your suspension there. So whenever you are preparing a cell, right, so you are provided with a suspension there. So basically they are saying that there are about 100,000 cells there. What is the maximum number of 18 millimeter round cover slips that can be plated at a density of 50 cells per millimeter square? So they are also providing that this is the density that should be placed within that cover slip there, and you can see that at 18 millimeter is also present there, which indicates the diameter of the cover slip there. So basically, what does mean by the cell density? Cell density means that if you are comparing a excuse me, sorry, I will just join right now. Okay, so I think that issue has been sorted out. Okay, I think this is fine. So you can base that based in the cell density. If you are comparing, if you are taking a 1 millimeter square or a particular area, this is the area of the cover slip that they are providing. A small 1 millimeter square is a really small area of that cover slip. In that area, how much of cell would be present there? The number of cells that would be present in that 1 millimeter square of that cover slip. So here it is given in the question. You can see that they have given us 50 cells per millimeter square, right? So if you're comparing 1 millimeter square, it has been said that there should be at the final, after the spotting or the incubation, there should be approximately 50 cells that should be present within 1 millimeter square of a cover slip there. So and also they have given that it is 80 millimeter in diameter. The length of the cover slip is about that. So from that you can find the radius, right? So if it is 18 millimeter, then it is the diameter. So radius is always half that of the diameter, right? So it comes out to be 9. So if you are calculating the area, total area of the cover slip, basically you know that it is round, right? They are given it as round. It is basically circular in shape. So in that case, you know the area of a circle there, right? So basically that area of the circle is pi r square. So if you know the diameter, you can calculate the radius from that, and then you just submit the value there, that is the 9 millimeter there, and pi, you know the value is 3.14. So in that case, if you calculate, you get the value of 254.34 millimeter square, which is the area of the total cover slip there. So we know the area of the total cover slip there, that is approximately 254. You can 254 millimeter square, which is the unit of that particular area there. So you know that particular value. So as I said, cell density represents the number of cells that are present within that particular 1 millimeter square of the cover slip there, right? So they have given that in 1 millimeter square of that cover slip there are 50 cells there. So you know that in 1 millimeter square there are 50 cells. You know the total area of the cover slip is 254.34 millimeter square. So in order to calculate the total number of cells that would be present if that particular cell density is 50 cells per millimeter square, and if you know the value of the total area of cover slip as 254, then just multiply that total area with this cell density. You get to know the value of the number of cells that are present there. Approximately 12,717 cells will be packed into that particular cover slip based on the information that is given to us. So if you know that in a single cover slip 12,717 cells would be added there. So you have given a suspension consisting of about 1 lakh cells. You have given that 100,000 cells are present there, or 1 lakh cells are present there. So in order to calculate the maximum number of cover slip, just divide this particular 1 lakh with 12,717, that would yield you the value of 7 cover slips. So basically that's the answer basically. So if I have a 100,000 cells in my hand, what is the maximum number of 80 millimeter round cover slip that can be plated at a density of 50 cells per millimeter square? And the answer is basically we are getting 7. So does anyone has any doubt regarding this?
No, sir.
Basically it's a mathematical question, right? So that's why I thought to write it down and then explain it to you. I hope that is understandable there. Okay, so I will just move on to the next question then. So coming on to question number 7. It's also a basic mathematical question. It's similar to the previous question, but some extra step is there basically. So scientist X has about 500,000 cells in 500 microliter as a cell suspension there. S decides to plate cells on 50 millimeter round cover slip at 500 cells per millimeter square. What volume or approximate volume should X add from the cell suspension per cover slip to achieve the desired plating density, given 1 ml is equal to 1000 microliter? So option 1 is 1 microliter, option 2 is 35 microliter, option C is 88 microliter, or option D is 127 microliter. What might be the answer? Anyone knows the answer? Or should I give some time to calculate so you can also get an idea about it? So I think I will just explain the answer here, how it was done and all. So it's a mathematical question, right? So it might take some time. So the answer to this question is 88 microliter. So I will just give a conclusion of how it was calculated there. Basically, you have from the earlier question, it was given that we have cell suspension there. There was 1 lakh cells. In this case, it is 5 lakh cells there, right? And you also know that they have given about 500 microliter. In that 500 microliter, 5 lakh cells are there, right? So and also they have given the diameter of the cover slip, and also they have defined the specific cell density that should be plated onto the cover slip, right? So what volume should X add to a cell suspension per cover slip? Earlier we want to find the number of cover slip that could be utilized into specific density, right? So in this case, what should be the volume that should be added from the cell, so in order per cover slip to achieve the desired plating density? They want to know the volume, not the number of cover slip. So basically in this case you can start with the same definition there. Basically you have the diameter there, from that you can calculate the radius. In that case 7.5 millimeter would be the radius there, and from the equation that pi r square, that area is equal to area of circle is equal to pi r square. So from that equation, if you just substitute the value, you will get the area of the cover slip as 176.7 millimeter square there. So it has been said that the cell density is there. So if you are comparing 1 millimeter square of that cover slip, 500 cells can be plated. It has mean 500 cells should be the maximum cells that should be plated there or present there. So in this case, you know the overall area of the cover slip. So in that overall area of cover slip, in that small area or 1 millimeter square area, 500 cells should be plated in this case. So if you are talking about the overall number of cells that will be present within that particular cover slip itself, it turned out to be like 176.7 millimeter square into 500 cells per millimeter square. So the total area, so total cells that would be present within that cover slip at the final spotting and all, you will get to be 88,350 cells there. So that much can occupy the cover slip on the base of that given cell density, that is 500 cells per millimeter square. So you know that answer basically. So you know that about 88,350 cells can be spotted in that cover slip there. So it has also been given that in the 500 microliter suspension that we have, about 5 lakh cells are present there, or 500,000 cells are present there. So I will just write it down. 5 lakh cells are present there. in that 500 microliter suspension. So if you want to take up one cell, you just divide this 500 with this 5 lakh cell. So if you are, so that will give you a value of about 0.001 microliter. So if you are taking about this much volume, that is 0.001 microliter or 1 nanoliter, one cell you will be able to pick out. So that what it means basically. So basically, and also you know that from the previous calculation, 88,350 cells could be spotted there. So if you just substitute this value here, 88,350 cells, then you multiply it with 0.001 microliter of the total volume, you will get that in order to achieve this particular cell density from this particular cell suspension. From this particular cell suspension, you have to take about 88 microliter, so that the whole cover slip can be spotted with that particular 500. So what you say? This 500 cells per millimeter square density of that cover slip itself. So that gives you the answer of 88 microliter there. So anyone has any doubt there? It's kind of a mathematical question. So if anyone has any doubt, you can ask basically. Okay, so I think this is quite clear, so I will just move on to the next question. So if you have any doubt, you can ask at the end of the presentation also. So coming to question number 8. So read the following statement. Glutamax has the same chemical formula as glutamine. Glutamax is used for the same purpose as glutamine in cell culture. Glutamax is an amino acid while glutamine is a dipeptide. Glutamax is more stable than glutamine. Choose the correct option. Only A is correct, only A and D are correct, only C is false, only B and D are correct, which may be the answer.
Option 4.
Option 4. Okay, so you are basically correct. Option 4 is the right answer. Only option B and D are correct. So basically, if you are, it is basically a chemical, right? Like it's a kind of a nutrient, right? Glutamax is a dipeptide form of glutamine there. So basically, glutamate has same chemical formula as glutamine. No, it's basically a dipeptide of glutamine. So it doesn't have the same formula as glutamine. Rather than it would just multiply it by 2. Then this chemical formula will change there, right? And then some water will be removed there, and etc. etc. So it won't have the same chemical formula as a glutamine. So glutamax is used for the same purpose as glutamine in cell culture. Yeah, basically it is right, because glutamine in its native form or individual form gets degraded easily. Basically you don't want that. So glutamax is basically a dipeptide there. So in the dipeptide form of when the two molecules of glutamine join together to form a dipeptide, they are more stable as compared to an individual molecule of glutamine. So that's why it's used as a cell culture as a nutrient component there. And glutamax is an amino acid while glutamine is a dipeptide. Basically it's the totally opposite, right? It should be like glutamax is a dipeptide while glutamine is an amino acid. It's totally the opposite there. So glutamax is more stable than glutamine. Yeah, that's right. That's why it is converted into a dipeptide, right? Rather than just providing it as an amino acid, it is used as a dipeptide because of the stability, the overall stability that it has as compared to this individual amino acid form. So in this case, option only B and D are correct. Others are wrong. So anyone has any doubt?
No, sir.
Okay. So I will just move on to the next question there. Question number 9. For monoculture, the cell yield is proportional to height of the culture, available surface area of the culture, available surface area of the flask, distance between cellular colony, or size of cell body. Which might be the answer? For monolayer culture, you have to understand what a monolayer is, then you will get the answer basically.
Option 2, sir.
Yeah, so you are basically right. So the answer is option 2, the available surface area of the flask. So there are different type of culture, like one is monolayer culture. If you compare, other one is multi-layer culture or a three-dimensional culture, right? 3D culture is also there. So monolayer culture is basically a culture that is grown at the top of the cell culture in a single layer there. So it is present as a single layer rather than a multiple layer there. So these monolayer are just if we found the appropriate extracellular matrix there, they adhere to it, then they form a single layer at the cell surface itself. So in that case, basically if you look at the monolayer culture, the as it forms a single culture, it just proliferate in a unidirectional way. It proliferate and spread in a unidirectional way, and then it covers the surface area of the flask there. So if you want to know how much cell yield it is, basically if you know the surface area of the flask there also and how much it has spread to, basically you can calculate the cell yield there, because it actually exists as a monolayer, right? If it is a multi-layer, then it's a different case. From that we cannot determine the cell yield there, because it grows as a single layer there, right? From the surface area that is available, surface area that is present within the flask, you can determine how much cell yield or how much it has the monolayer culture has actually divided as well as proliferated or differentiated, whatever it may be. So for monolayer culture, the cell yield is directly proportional to the available surface area of the flask, how much surface area is present there rather than it getting spread there. So does anyone has any doubt there? Okay. So I think this is a basic question, right? So monolayer culture and everything. So I will just move on to the next question then. Coming on to question number 10. Read the following statements carefully. Animal origin free media, about it is animal, it is related to something like animal origin free without a media which doesn't consist of animal, anything that is obtained from an animal source there. So first option is not manufactured using any raw material ingredients derived from an animal, including human tissue, cell or body fluid, that is at a primary level of manufacturing. No animal derived materials are used as a starting material or in the primary level manufacturing process. The components used to manufacture the raw material ingredients are not derived from animal tissue, cell or body fluid, that is at secondary level of manufacturing. Ingredients and components may be produced by or extracted from plants, bacteria or yeast or cell-free system derived from them, or may be produced by organic synthesis, may contain recombinant animal or non-animal proteins, provided they are expressed in plants or microorganisms. Choose the correct option, which might be true. Only A is true, only B is true, only C and D are true. A, B, C, all of them are true basically, which might be the option. Just read the option and then tell me the right answer. Animal origin free media, basically it doesn't consist of any animal source and all. Directly from any animal source they don't have.
Option 4, sir.
Option 4. Yeah, so you are right. Basically, option 4 is the right answer. All of them are true. That is A, B, and C, and D are true. Basically, if you look into animal-free media, you can see so that it doesn't use any animal source from there. So basically, if you look into the first option there, is not manufactured using any raw material ingredient derived from animal source. No animal derived materials are using as starting material or in primary level of manufacturing. So that option is right, because they don't probably have any animal source in there. So basically from the name itself is quite clear. Option B, so the components used to manufacture the raw materials are not derived from animal tissue or a body fluid. So basically that gives you the same definition there. So that option is also true, that is used to, that are not derived from any animal source. And ingredients and components may be produced or extracted from plants. So it has been said that animal free media, right? Animal origin free media. It doesn't mean that we cannot use other components like that are obtained from this particular plant, bacteria or yeast. So that might be produced, or anything that is produced by organic synthesis, that would be present within this particular media. So only animal sources are not allowed in this media. So this option is also right. So may contain recombinant animal or non-animal proteins, provided they are expressed in plant or microorganism. So animal sources directly which is obtained from animals are not there, but something that is indirectly present within animal source can be used there, like recombinant animal or non-animal proteins. Suppose that if you are if you are cloned a particular gene that is present within the animal source there, or anything that is present with the animal source there, and then you are expressing that particular protein in bacteria or plants there. So you can use that because you are not directly obtaining that from animal, but you are basically obtaining the signature or something that dictates that animal protein there and then expressing it out, right? So that particular protein or any other component can be used within that animal origin free media there. There is no problem regarding that kind of usage. So this option is also right. So in this case all the options are true. A, B, C and D are true. So does anyone have any doubt regarding this? Okay, so I think that's quite clear. So I will just move on to the next question. So coming on to the next question, question number 11. What is the character shape of the hippocampal neurons? Round, pyramidal, elongated or hexagonal? This might be the answer.
Option 2, sir.
Option 2. Yeah, you are right. Option 2 is actually pyramidal in nature. This is a diagram there. You can see that this is the cell body there. I think it's called a neuronal body or cell body there. You can see that the specific pyramidal shape that is possessed. You can see the dendrites that are present there, and also you can see the synapse there, right? So basically some proteins that target certain synaptonemal proteins, what it is, synapse proteins like synapsin and all, can be used to fluorescently brighten it out. Some antibody that binds this protein, then you are tagging that antibody with a particular fluorophore there. You can basically give out fluorescence there. So you can see the entire structure of this hippocampal neurons there. So basically, if your hippocampal neurons are usually needed for memory formation, learning, as well as understanding the navigation, what direction you are going through and everything. So this plays an important role. So in case of memory, as I said, memory formation can be used to related to both short-term as well as long-term memory there. And it is basically used in case of studying neurodegenerative disorders there. So if you look into hippocampus and all, because it is associated with memory, if this particular hippocampal neuron gets degraded out due to accumulation of proteins like what is it, tau proteins are there, then it can lead to protein accumulation, which can then lead to this neuronal death and all. So subsequently when the neuron dies out, then chances are that you will develop this Alzheimer's disease and everything, or a neurodegenerative disease as widely used term there. So as it is associated to memory there, right? If memory is lost there, people cannot recall anything basically there. So that's why these hippocampal neurons are like in a much important field of study there. So does anyone have any doubt regarding this? I think this is clear. It's just a basic question, right?
No, sir.
Okay. So I will just move on to the next question then. Coming on to question number 12. Sorry. So papain is a I think this have been repeated. So papain is a which enzyme derived from where? So first one is proteolytic enzyme papaya, proteolytic enzyme milk, lactate dehydrogenase papaya, lactate dehydrogenase milk. Which is the answer?
Option 1, sir.
Option 1. So papain is basically a proteolytic enzyme that is derived from papaya, or scientific name Carica papaya, right? So this is a cysteine protease that is used to degrade out extracellular matrix. So if you are doing a cell incubation and all, you have to separate the tissue from cells from the tissue, right? So papain you are adding at a specific low yield concentration, and then it degrades out all these extracellular matrix proteins that are present without affecting the cell viability, and followed by, if you are doing a titration or a mechanical dissociation there, you are doing it using a pipette tip, and then you are plating that it in a particular cell culture plate or a cover slip, whatever necessary, pouring out the media, and then you are incubating it out, right? So basically papain is used in this enzymatic dissociation of this cell from the tissue, which breaks the extracellular matrix proteins and all. So I think that particular answer is clear, and this is a structure of the papain there, which has been obtained from PDB. So I think I will just move on to the next question then. So coming up to question number 13. In a hemocytometer, volume of 1 millimeter into 1 millimeter square is, which might be the answer. They are asking about the volume. So in this case, this is a hemocytometer. So basically they want to know if you are adding cell culture into or a particular cell suspension into this particular chamber there, how much volume can it occupy? You know what?
Option
Yeah, go.
Option 4.
Yeah, so the answer is option number 4, 100 nanoliter there. So basically this is a hemocytometer. You can see that there are 4 grids there. So in some cases there are 2 grids there, and depending on the hemocytometer that you are using, it might change. So basically hemocytometer is a type of a cell counter, right? So basically there are manual type of cell counting as well as automatic type of cell counting. So if you are using optical density or any electrical impedance to calculate the cell count and all, then it is a type of automatic cell counting there. So in case of hemocytometer, it is a manual cell counting. You have to pour a cell suspension. You can see that this is hemocytometer there. You are placing this type of grid at the top of this, and then from the side using a pipette you are actually pouring out the cell suspension there. So based on the volume that it can occupy, it will actually absorb that cell suspension into it, and when after that you will look into a microscope there. So based on the grids that are present there, individual grids, you calculate the number of cells there, and then you get an average value there. So based on that, you can calculate how much cell that is present within that total volume of suspension that you have. So the answer is 100 nanometer. So basically, if you look into this structure, you can see that there is 1 millimeter into 1 millimeter square, right? So this total square is 1 millimeter into 1 millimeter, that is length into breadth that you have given. And if you look into the height of that molecule, it is consists of there is a small space there, so that the cell suspension can actually go into. So if that space might not be there, then cell suspension won't be absorbed into the hemocytometer. So a small opening is there of about 0.1 millimeter, so that you can pour the cell suspension into it. So basically the dimensions are 1 millimeter into 1 millimeter into 0.1 millimeter, which actually represents the depth. So this actually represents a cuboid like structure, right? Rather than a cube, it represents a cuboid like structure in 3-dimensional. So volume of a cube, you know that the volume of a cube is length into breadth into height. So you know that the length is given as 1 millimeter, breadth is given as 1 millimeter, and the height or the depth is 0.1 millimeter. So if you just give the value, in this case you get the value as 1 into 1 into 0.1, that is 0.1 millimeter cube, or millimeter cube actually is equal to 1 millimeter cube is equal to 1 microliter. So if you just change the unit there, you will get as 0.1 millimeter cube is equal to 0.1 microliter. Or if you compare into the units that the option is given, that is nanoliter, you will get the value of 100 nanoliter, because 1 microliter is always equal to 1000 nanoliter, right? So in this case 0.1 is there, 1 decimal point is shifted, so you will get a value of 100 nanoliter. So I think this answer is clear. Does anyone have any doubt? So this is a basic question basically. So I think the answer is clear. So I will just move on to the next question. So coming on to question number 14. Which of these antibody is used to label mostly neuronal cell? First one, option number MSC, MAP2, islet, or anti-RYR. Which might be the answer? Labeling neuronal.
Option number 2, sir.
Option number 2. Yeah, you are right. Basically, MAP2. So basically, if you give a definition of all this, MHC, you know that it is also called as major histocompatibility complex, right? Basically, immunoglobulins have this particular type of MHC complex there. So it is involved in immune response and not specifically present in neuronal cells. So it cannot be used for labeling and all. Sorry, neuronal labeling and all. MAP2, also called as microtubule associated protein 2. Basically, I think the abbreviation is not there. So basically it is forms a particular component of this neuronal cytoskeletons there. So it is expressed in these neurons and is widely used to identify neuronal cells in various studies, especially immunofluorescence. Basically, and basically from the you can know that basically why we are tagging a particular protein with antibody, right? So if you have a particular antibody that can bind against this MAP2, which is present in neuronal cell, you can basically incubate this cell with this antibody for some time, and then introduce a secondary antibody which is tagged with a fluorophore there that binds to the primary antibody there. So this gives us a particular fluorescence there. Basically, if you I will go into one of the previous questions there, right? So you can see that, right? Suppose that the map to this green fluorescence protein, suppose that it recognizes the map2 proteins there. So secondary antibody actually lightens up the particular position or the location where this particular protein is present there. So this is basically called as immunofluorescence. Basically, using an antibody that targets a particular protein there, you are actually using that particular mechanism to visualize the or identify the location of a particular protein there or any other substance there. So this particular technique is called as immunofluorescence, and it relates the presence of a or a use of a immunofluorescence or a fluorescence microscopy there. So yeah, so that's why MAP2 is used in this case. So islet is basically cannot be used because it is present in insulin producing cells there in pancreas and all. So and hence it is not present in neuronal cell and cannot be used to label or study neuronal cells itself. This anti-RYR or ryanodine receptor, it is actually used in calcium signaling and is found in muscles and some neuronal cells, but not mostly in case. But if you want to study mostly neuronal cell, you have to study a protein that is predominantly abundant in neuron itself, right? So we basically cannot use this in this case also to label this neuronal cell. So answer to this question is which of the antibodies used to label neuronal cell? MAP2 is the answer. So or microtubule associated protein 2 is the answer. So basically, if you look into some of the functionality of this MAP2 protein, you can see that it is used in labeling neuronal cell as you have studied that, as it is a part of a neuronal cytoskeleton there. And it is used in cytoskeletal stabilization, especially to maintain the cytoskeletal integrity there. Microtubule dynamics, basically it binds, it has capability to bind to MAP2 has the capability to bind to several microtubules that are present within the neuronal cells there. So and it helps in the assembly of these microtubules, and hence it helps in this microtubule dynamics and all. So and also it is involved in signal translation because MAP2 is a protein that helps to anchor protein kinase A to the membrane. So protein kinase A is actually involved in signal cascading and all, so it is involved in signal transduction and disease pathology and all, because the level of MAP2 somehow dictates to the level how whether any neurodegenerative disease like Alzheimer's is present there or not. So it is somehow related to this MAP2, this disease pathology or neurodegeneration and everything. So answer to this question MAP2. So anyone has any doubt regarding this? Okay, so I think this question is also so quite clear. So I will just move on to the next question then. Coming on to question number 15. Which of these is not a developmental stage in rat? First one, embryonic, prenatal, rental or neonatal. Which might be the answer?
Option 3, sir.
Option 3. So basically embryonic stage, you can see that the answer is right. Option 3, rental. So basically all the other options are involved in some kind of developmental stage in rat. So embryonic stage, you can see that this is a developmental stage that is found within the embryo, within the uterus itself. And prenatal, basically before giving birth to that particular organism, that is a particular stage which is represented as a prenatal there. Natal means birth, prenatal means before birth there. So it is somehow related to this embryonic stage because it is related to a period that is of the fetus that is present before the birth. And in case of neonatal, it is the early life of rat. It is related to something that comes after giving birth to that particular organism. So this stage pertains to the earlier life of rat, specifically after birth. And I think rental is no kind of a embryonic developmental stage or a developmental stage in rat. All the other steps, that is embryonic, prenatal as well as neonatal, corresponds to the developmental stage in rat. So I think that answer is also clear. So I think I will move on to the next question. Does anyone have any doubt regarding this?
No, sir.
Okay. So coming on to question number 6. Which of this protein misfolding and aggregation, which of protein misfolding and aggregation is linked to Alzheimer's disease, which is a neurodegenerative disease, right? So option number 1, amyloid beta, tau, both A and B, none of A and B. Which might be the answer?
Both A and B.
Both A and B. So basically you can see that both A and B, you are right. Basically both amyloid beta as well as tau proteins are involved in neurodegeneration, including Parkinson's as well as Alzheimer's disease. So basically these are basically protein aggregation. So as I said earlier, that protein aggregation can lead to protein accumulation can lead to unwanted protein aggregation within the cell. So if protein gets aggregated above a particular limit, the tendency of that particular proteins to cause cell death is much more as compared to in normal state. So amyloid beta is some kind of a kind of that particular protein aggregation, especially protein plaque formation there. So basically amyloid, in case of, you can see that from the picture that these are amyloid beta plaques, right? So these are proteins that aggregate together and forms a plaque there, and this in turn accumulates within the cell and leading to a formation of neurofibrillary tangles there, because the protein cannot, so the neurons cannot assemble itself for the over the extracellular matrix because of the accumulation of these proteins there. So in that case, when these proteins get accumulated over time, the chances are that these neurofibrillary tangles will be forming more, and this lead to cell death there. So amyloid beta is one kind of kind of this particular plaque formation or accumulation there that is hallmark of Alzheimer's pathology there. And basically this is insoluble in nature, that's why they are aggregating together to form, and then it leads to neurodegeneration there. And coming on to a tau protein there also, you can see that it is also implicated to neurodegeneration disease like Alzheimer's or Parkinson. So this tau is something that stabilizes the it is a protein that stabilizes these particular microtubules there, because microtubule assembly is an important function in case of neuronal cells and all. So if it is somehow affected there, or if the stabilization mechanism is somehow lost there, then it can lead to disease condition and all. So it stabilizes the microtubule, which is essential for maintaining the cell structure as well as the cell function. However, in case of Alzheimer's disease, this tau or these tau proteins, right, these get hyperphosphorylated, and this leads to neurofibrillary tangles there. In case you can see that the neurofibrillary tangles are there, in this case also you can see the formation of neurofibrillary tangles there. So this tau gets hyperphosphorylated there, and this forms neurofibrillary triangles within the neurons, and this in turn leads to neuronal death there because of the aggregation of tau, and this leads to disease conditions like Alzheimer's or Parkinson's or other neurodegenerative diseases. So this is just a brief mechanism of how protein accumulation happen and leads to cell death. So I think this question is clear. So I will just move on to the next question. Anyone has any doubt regarding this? Okay, so I think this question is quite clear. So I will need to move on to the next question. So question number 17. Which of this brain region is associated with memory and learning? First option, hypothalamus. Second option, amygdala. Third option, visual cortex. Fourth option, hippocampus, which might be the answer. We have discussed this earlier.
Option 4, sir.
Yeah, so the answer to this question is option 4, hippocampus. Basically, these are neurons that are present. These are hippocampus consists of several neurons, right, which are pyramidally in origin. And these neurons have the essential property of converting short-term memory into long-term memory, because in order to store that particular memory in our brain, then the short-term memory needs to be converted into long-term memory which afterwards we can recall out, right? So it organizes these stores and retrieves memories, allowing individuals to recall information whenever needed. Both short-term memories are so in this mechanism, both short-term memories as well as long-term memories are stored in this case. So apart from that, it is also involved in recognizing spatial navigation, because in order to move from one place to another, right, in order to familiarize with the place, you need to understand the environment first, or the surrounding atmosphere first. So spatial navigation plays an important role in this, and this particular function is regulated by hippocampus itself, because you need to remember the location where you have kept one object, or if you have to move from one place to another, you should know the way to that place, right? So this all comes within the memory there, memory storage. So hippocampus helps to store this memory, and because of the memory recognition function of this hippocampus, it also helps in spatial navigation and all. And also memory representation, you can see that this allows individual to adapt their behavior based on past experience and social interaction, which is crucial for effective decision making and social functioning. So this portrays to learning mechanisms. So from memory representation or memory that you have stored within your brain, what you learn from that. So that actually is also a function of hippocampus there. So this is why hippocampus is required for not only just memory function, regulating memory functions, but also in regulating the learning function of an individual itself. So hippocampus also, amygdala also have some function, but most probably the important function of this particular memory and learning is done by hippocampus rather than amygdala. It actually closely works with amygdala to link memory with emotion, and this encoding emotionally significant events helps to make it easier to recall memories through recall things later through the memories that are present stored within our brain. So this is the answer. So which part of the brain is associated with memory and learning? So the answer is hippocampus. So anyone has any doubt? And a visual cortex is used to study this visual representation that you are getting from your eyes, right? And hypothalamus, you know that it is used to control the hormonal regulation within the body, as well as maintain the body hemostasis, as well as the body temperature regulation and everything. So it is not regulated with the memory and learning. So hypothalamus is involved in memory and learning rather than the other three options there, apart from amygdala. So I will just move on to the next question there. Coming on to question number 18. Neurobasal media is buffered by first option, basic amino acid, oxygen, sodium bicarbonate, or glucose. Which might be the answer? Neurobasal media.
Sodium bicarbonate.
Sodium bicarbonate. So we have discussed that, right? So basically all the components are present there. Basically amino acids are there. In some cases glucose is there, more than the sodium pyruvate is present, provided there. So sodium bicarbonate is the neurobasal media is buffered by sodium bicarbonate. So basically neurobasal media is a specific media or a defined media that is used to grow or culture neuronal cells only. So there are different types of neurobasal media there. Basically B27 is there. One example is B27, which consists of a commonly used neurobasal media there. It is used to grow this prenatal as well as fetal neuronal cultures there, and it consists of low osmolarity there. It also supports the long-term maintenance of embryonic neurons there. Basically the majority in cases, if you want to study a neuronal culture, the neurons are basically obtained from rat hippocampus, their fetal rat hippocampus. So basically the embryo after 18 days of gestation, like when the embryo is formed after 18 days, the fetus is isolated from the mother is sacrificed, then the fetus is isolated, then the brain is removed, and from that hippocampus is dissected out using surgery, right? Hippocampus is dissected out using surgery, right? So basically all this media, that is neurobasal media, is used to grow this fetal neuronal cell, not adult cell. And one such example is B27, and this 27 represents the number of components that are present within this neurobasal media. Neurobasal A media is a specific type of media that is used to grow adult neuronal cell, not fetal neuronal cell. And it consists of high osmolality there. Basically, in the presence of sodium chloride, level of sodium chloride is small, which might be changing the osmolality level there. Because for growing fetal neuronal cell, in comparison to adult neuronal cell, the osmolality of fetal neuronal cell or the media that is required is 215 or 220 milliosmoles there. In case of neurobasal A media, which is used to grow adult neuronal cells, the osmolality is much more higher there. It goes up to 260 millimosmoles there. So there is a difference of osmolality that is required to grow these two types of neuronal cells, right? So osmometer is used to calculate the osmolality of that particular media, and also since because of that difference, these two different types of media is there. Commonly used is neurobasal media and neurobasal A media used for growing adult neuronal cells there. And if you are looking into what is it? The buffering capacity sodium bicarbonate, you can see that if the pH is actually getting higher within the media due to the metabolic release of protons or any other components there, then this sodium carbonate releases this bicarbonate ion that combines with protons present within the media, which has been produced excess in the media, thereby making the media optimum at a pH range of about 7.2. Sodium bicarbonate is actually supplemented within the media to counteract the pH level changes there when you are actually doing a cell culture, and because of the metabolic reactions and everything taking place, this pH will fluctuate there. So sodium carbonate is used to buffer this particular pH fluctuations there. So I think the answer is clear. So anyone has any doubt? Okay. And also phenol already you know, and sodium pyruvate is also used for, it is also a free radical scavenger there, and also it can be shunted into like this which one, like after the glycolysis and all, it can shunt into different pathways required for energy formation and all. D-glucose is used for glycolysis and all. So that's the energy providing source there, right? And also 5% carbon dioxide is also used because in order to maintain the buffering capacity of the cell there within the incubator there. Yeah. So I'll just move on to the next question then. Read the following statements carefully. Xeno-free media. A. Option does not contain ingredients derived from non-human animals, does not contain recombinant materials made from non-human animal DNA sources, may contain purified, processed or unprocessed material from human sources, may contain recombinant materials made from human, plant, bacterial or yeast DNA sources, sequences. Animal derived components may have been used as raw materials at a secondary or tertiary level of manufacturing unless otherwise indicated. First option A is false. Second option D is only true. A, B, C are true, and last option all are true. Which might be the answer? Xeno-free media.
Option 4, sir.
Option 4. All the options are correct, right? So basically, xeno-free media is a media which can use some components that are derived from humans, but it cannot derive ingredients that are found that are obtained from non-human animals. Other than humans, they cannot use basically. Or plants like bacteria, yeast DNA sequence they can basically use, or any other source you can use, but they cannot use it in components that are derived from non-human animal source. So first option coming does not contain ingredients derived from non-human animals. Second option that is basically correct, right? So basically that's the definition of the xeno-free media. Second option does not contain recombinant materials made from non-human DNA sources. That's also true because you cannot, even in case of like animal-free media, it is not similar to animal-free media. But in case of xeno-free media, you cannot even use any other recombinant material that has been obtained from non-human animal DNA sequences, and may contain protein, purified, processed or unprocessed material from human sources. Yes, it can include sources that are obtained from humans itself, so there is no issue with using this. And may contain recombinant materials made from human plant, bacterial, or yeast DNA sequences. Yes, other than humans, non-human animal source cannot be used, but other than other components like humans, can include components, plant, bacterial, or yeast DNA sequence. They can also include, no issue in that case. So animal derived components may have been used as a raw material at secondary or tertiary level manufacturing unless otherwise indicated. So it's not mandatory, but if you are providing anything from outside, you should have a proper designation or indication regarding that this is provided there. So yeah, so in this case, all the options are true. That is, all the options are true in this case. So does anyone have any doubt regarding this? Okay, so I think it's quite clear. So I will just move on to the last question there. So yeah, coming on to question number 20. Leibowitz L15 media contains dash and but lacks dash. First option is sodium pyruvate, sodium carbonate, sodium tartrate, sodium bicarbonate, sodium glutamate or sodium bicarbonate, sodium pyruvate or sodium bicarbonate, which might be the answer. L15 media. This we have discussed in the first week itself.
Option 4, sir.
Option 4. Yeah. So the answer is correct basically. Leibowitz 15 or Leibowitz is the name of scientist that actually made this media basically. L15 media contains sodium pyruvate but lacks sodium bicarbonate. Basically, if you look into the sodium bicarbonate buffering and all, it's not actually a good buffering system because the pH actually fluctuates at a much higher range than it could actually maintain. Basically, it is regulates the pH, but actually the fluctuations are much higher as when you are using this particular bicarbonate buffering system and all. So this particular media actually lacks this what you say, this sodium bicarbonate there, right? So if sodium bicarbonate is absent there, and if you are growing the media in an incubator which actually lacks the carbon dioxide that is provided from outside, that is, we are basically growing in a normal incubator there other than a CO2 incubator, because CO2 incubator is what needed if you are using sodium bicarbonate in the media in order to regulate the level of pH there, right? So if you are not if you are growing this particular culture or a cell culture in Leibowitz 15 or L15 media, then you probably don't want to grow the cell culture. You probably don't need to grow the cell culture in that particular CO2 incubator. You don't need to provide the CO2 from outside before the buffering is known, because it does not depend on this particular type of buffering there. So L15 is buffered by other components, other complements of salt, free basic amino acid, and galactose. Sometimes glucose was also used, but in this case galactose is used to maintain the physiological pH control there. So based on the combination of all these components there, the pH is maintained there, rather than providing this carbonic acid buffering system. Based on the complement of this particular amino acid or a composition of this specified composition of this amino acid, salt and galactose, the physiological pH is actually regulated there. And apart from that, it also contains 2 millimolar L-glutamine, sodium pyruvate, and no sodium carbonate, as we have discussed before, because it does not depend on carbonic acid buffering. Sodium pyruvate is also used there because it has a free radical scavenger, and it also can be used as an energy source to produce ATP and all, and facilitates and maintains the cell culture even in the absence of an incubator. CO2 incubator is not, a normal incubator is needed, but a CO2 incubator is not needed in this case to maintain the the growth and incubation as well as the growth and proliferation of the cells there. So that's what Leibowitz media contains. Leibowitz media or L15 media contains sodium pyruvate, but it lacks sodium bicarbonate because it does not depend on carbonic acid buffering and all. So anyone has any doubt regarding this?
No, sir.
Okay, so I think we will end the class today for this, based on this. So anyone has any doubt regarding the overall class that has been taught today? Anyone has any doubt? Okay, so I think if you have any doubt, you can directly message me, or in the next class you can also ask the question. No issue in that case. So thank you everyone for joining for today's class. I hope you will all join for the next class also. So thank you.